Okay, so we're going to talk about um, one more concept before we move on to tissues. And I want everybody to really pay attention to this unit because this is something that if you don't really have a concrete understanding of it, just the basics, then you're really not going to understand the bigger stuff that we go over throughout the course. Um, and when I took anatomy and physiology, um, it was later on in college. I didn't take it in high school. Um, but this is one thing that I wish I had a better understanding of before I went into anatomy and physiology, because it would have helped. It would have been a lot easier on me. So here we have, we're going to talk about the plasma membrane, and that's this guy right here. And many of you probably know the plasma membrane by a different name, the cell membrane. Plasma membrane is just a fancy name for it. It separates the living cell from its non-living surroundings. This is a double-layered, thin barrier, controls traffic into and out of the cell, and is known as the lipid bilayer. Like other membranes, the plasma membrane is selectively permeable, meaning that it's semi-permeable, allowing small, uncharged substances to cross more easily than others. And all semi-permeable means is that it allows some things in and allows some things out, but it doesn't allow everything in. It doesn't allow everything out. It's selective about what it lets in and it lets out. We call this the plasma membrane because it consists of something called the phospholipid. And a phospholipid is just these little guys right here. They consist of, this is, they have a head and they have a tail. Now their tails are made up of um, fatty acid chains or lipids. And if you remember from biology, lipids are supposed to be, they can't mix well with water, okay? So the tails do not mix well with water. And then they also have a head, which is made up of phosphate group, and it loves water. It can mix with water. And here we see it mixing with the extracellular fluid. And also the inside of the cell, which are composed mostly of water. Proteins serve, we also have proteins in here. Those are these purple things right here. And they serve as support and transport, and as transport molecules. As you can see, this one has an opening that allows into the cell or outside the cell, so certain molecules will be able to use that to come in and out. And there are several different mechanisms that they can use to come in and out. Depends on what type of transport protein that is. And we'll talk about that more later. Carbohydrates, they help in cell-to-cell communication, and they act as radio towers. So here we have these carbohydrate chains right here, and they'll help in cell-to-cell communication. They'll be like, oh, hey, I'm a red blood cell, things like that. Okay, so crossing the cell membrane. We're going to talk about two types of, or two ways that they can cross. They have passive transport, which is the movement of materials into and out of the cell that does not require energy. And then we also have active transport, the movement of materials into and out of the cell that requires energy. And materials move against the concentration gradient and move from low to high concentrations. So here we have the types of passive transport. We have diffusion, osmosis, and facilitated diffusion. And I'll talk more about this active transport a little bit later. So here we have, we're talking about diffusion, osmosis, facilitated diffusion. When I tell my students a way to remember passive transport, remember it as, riding your bike downhill. Because when you passively are riding your bike downhill, you're not using any energy, and you're going from a high concentration to a low concentration, okay? Or you're going from high to low. You're coasting downhill. Doesn't require any energy to coast down that hill. And when I'm talking about high to low concentrations, that's exactly what's happening here, guys. Here we have diffusion. This is the movement of molecules that can easily move in and out of membranes from areas of high to low concentration. It occurs when molecules move randomly away from each other in a liquid or a gas. And when we talk about this, we're talking about uh, diffusion when it moves down the concentration gradient, okay? Okay, sorry, I had to pause that for a second to plug my computer, so I'm going to maybe recap on some things that we just talked about. Diffusion, this is the movement of molecules that can easily move in and out of the membranes from areas of high to low concentration. It occurs when molecules move randomly away from each other in a liquid or a gas, and it moves down the concentration gradient. Now, what does all this mean? It means that we have areas of high concentration, so like diffusion, we could be talking about gases, we could be talking about perfumes that we spray into the air, or we could be talking about air fresheners. If you guys have ever noticed, if someone in the room sprays perfume, you might not be able to smell it at first if you're far away from them, but pretty soon that perfume starts to diffuse throughout the room, and pretty soon the whole room starts to smell like that perfume. Or air freshener. If you've ever sprayed it, in one part of the house, and all of a sudden you're smelling all over the house, it's because that air freshener is diffused throughout the house. One of my favorite examples of diffusion is, waking up to someone cooking breakfast down in the kitchen. Uh, you might not be real close to the kitchen, and the house certainly didn't smell like it when you went to sleep, but someone started making maybe some bacon or brewing some coffee, and all of a sudden those molecules move from an area of high concentration in the kitchen to lower concentrations throughout the house, and pretty soon it crept into your room, and you were waking up by the aroma of bacon or coffee or whatever, might be your favorite breakfast food. It occurs also with liquid, when like if you put a few drops of dye into water of a beaker of water, all of a sudden you would start to see those dyes diffusing throughout the water, or they would start spreading throughout the water. We call this moving down the concentration gradient, okay, or moving with the concentration gradient. When we're moving from high to low, we're moving with or down the concentration gradient. So here we have again, we're starting off with a high concentration spreading throughout maybe the house or whatever you liked better, like cologne, perfume, or the spraying of a skunk moving through the air. So those are some great examples. Okay, osmosis. This is a little different from diffusion. When we talk about diffusion, it can be any molecule that we're talking about here that diffuses easily. So we're just talking about oxygen or carbon dioxide, or we could be talking about it diffusing through a liquid or a gas, or diffusing through the cell membrane. But when we talk about osmosis, remember, this is very different. We're talking about the diffusion of water specifically. And we're also talking about it specifically moving through a semi-permeable membrane, okay? So you have to have those two things present for it to be called osmosis, all right? And water moves from areas of high to low concentration. So if we have an area of high concentration, it's going to naturally want to move to an area of lower concentration. Water moves down the concentration gradient, and this is a type of passive transport, so it does not require energy. There are three types of osmotic solutions, and we're going to talk about them a little bit now. There's hypotonic, where the cell will gain water, swell, and burst. Isotonic, where the water molecules move at equal rates from one to the other with no net osmosis. And then there's hypertonic. The cell will lose water, shrivel, and probably die. So hypotonic solution. This has more solutes and less water on the inside of the cell and less solutes and more water on the outside of the cell. Now that might seem a little confusing to you right now, but hold on, I've got a diagram. I want you to remember it this way, Hypo Hippo, because it causes water to move into the cell and swell like a hippopotamus. Distilled water is very hypotonic because it has no solutes, okay? And what that means is it's the purest form of water, all right? By now you should know what a solute is versus a solvent. In a solution, you have a solvent and you have a solute. A solvent is like water. A solute is going to be like salt. Whatever you're mixing is your solute. And the solution would be salt water. So solutes are anything that's mixed into a solvent or water. Hypotonic solutions are very bad for animal cells, but very good for plant cells. Plant cells will not burst when placed in water because of the cell wall. In fact, plant cells like lettuce must keep wet in order to be crisp. The pressure exerted from the cell wall is called turgor pressure. And I'm not going to talk too much about plant cells just because we're talking, this is anatomy and physiology, and we're mainly talking about animal cells. So here we have a hypotonic solution. Here's a solute. All this free space right here is water, and here is the inside of the cell. If you will look and see, there's way more water on the inside of the cell, or I'm sorry, the outside of the cell than on the inside of the cell. And like I said before, osmosis moves from areas of high concentration, or water moves from an area of high concentration to low concentration. So it's going to move into the cell, and it's going to make this cell swell up big like a hippopotamus, okay? So the greater solute inside, then this should say outside. And water moves in, solute moves out, cell swells. And the solute may not move out. It could move out, but it may not. It depends on how it's transported through the cell. Hypertonic solution. When the solute is higher outside of the cell than the inside of the cell, it causes water to move out of the cell. And if the cell loses too much water, the cell will shrink, shrivel up, and die. Cells in hypertonic solutions die because the activities are disrupted by the lack of water. So we need water to carry out our normal cellular activities, and if we don't have that, then cells die. Example, drinking salt water, bad for both plant and animal cells. And we're over here, we're talking about hypotonic solutions, and as you can see, we're talking about hypertonic solutions, and as you can see, there is way more water on the inside of the cell than on the outside of the cell. So it likes to move from an area of high concentration to low concentration, and boom, it moves out of the cell. Isotonic solutions. This is where the solute is equal both inside and outside of the cell. Molecules still move in and out of the cell, but there is no net movement. Same amount that leaves enters. Saline sprays and IVs are isotonic because they contain solutions isotonic with the human body to help replenish cells without offsetting homeostasis. And I'll talk a little bit more about these examples down at the bottom when we get to a different slide. But this is isotonic. Just a little hint, whenever you see ISO, I-S-O in front of a word, that means equal. So if you're talking about an isotonic solution, that's equal on both sides. So here we have no net movement of water is moving out. We still will have water molecules moving in and out, but they'll be moving at equal rates, okay? So like 2 water molecules might move out, 2 water molecules will move in. Salt. Here we have what I like to, we talk about a lot of, okay, if we place a cell in this water, but we don't talk about, okay, so here we're talking about, we're going to talk about if we have a hypotonic solution, a hypertonic solution on both sides. And we have the same amount of solution on each side, but this one has less solutes and more water, and this one has more solutes and more water. Water is going to move from the area of high concentration to low concentration. And if you can see, it's doing the same thing over here. Or this is afterwards. And you'll see they'll kind of even themselves out in concentration. Here we're talking about hypotonic solutions, isotonic solutions, hypertonic solutions. Okay, here are our animal cells up here. And here's a hypotonic solution. If we place a hypotonic solution with a red blood cell, this is what these are, red blood cells. Then what's going to happen is the cells are going to swell and burst. And the reason for this is it's not like a plant cell where it has this cell wall protecting it, but it's also because this lots and lots of water is going to move into the cell, and it's going to make it burst. Normal, we're going to have an isotonic solution. These are like our saline sprays and our IVs. These are isotonic. Those solutions are isotonic with our red blood cells, and they're not going to offset our homeostasis. So when we go to the hospital, they'll put in an IV to help keep us hydrated and to also deliver medications through. And that's because that those IVs have a isotonic solution. Here we have hypertonic solution. We also don't want to add too much salt water or we don't want too much salt because they'll make it shrivel up. I don't know what. Okay, now let's talk about facilitated diffusion. This is the movement of molecules across the cell membrane using carrier proteins or transport proteins. A type of passive transport moves from areas of high to low concentration. It moves down the concentration gradient. Most molecules are too big to cross the membrane by simple diffusion and require the help of protein carriers. An example is glucose enters and leaves the cell by facilitated diffusion. In facilitated diffusion, we're still moving from an area of high concentration to low concentration. We're just using a protein to help us get there. See, these guys are too big to fit through that cell membrane, so they've got to use a protein carrier. And here it's just another example. High concentration to low concentration, not using any energy. Okay, now we're going to talk about the other type of transport through the cell membrane. This is called active transport. A couple of things just real quick. Osmosis is really important when we start to talk about our urinary tract. It's also really important when we hold on a second. Okay, and then when we talk about active transport, we're talking about more. It's really important we start talking about the heart and nerves. A lot of that is managed through active transport, and a lot of it's managed through ion channels, which we're not going to talk a lot about right now, and that's okay because we're going to talk a lot about them later. So here we have active transport. It's the movement of molecules against the concentration gradient. It moves from an area of low concentration to high concentration. So we're doing the opposite of passive transport. It needs energy and usually requires the use of protein carriers that pump materials across the cell membrane. Okay, a lot of times when we talk about active transport, you're going to hear a lot more about protein pumps or ion channels than you are going to hear about bulk transport, which we're going to talk more about in this PowerPoint. You'll hear a little bit about it, but we're going to talk, just give the basic foundations of it, the protein carriers now, and then we'll move on to transport. So here we have molecules move up or against the concentration gradient from low to high. Okay, so you're going to move from an area of low concentration to high concentration, and you're going to use a lot of energy to do that. That's kind of like riding your bike uphill, because you're going from an area of low concentration to high, and you're using lots and lots of energy to pedal up that hill. Bulk transport. This is another type of energy that is not the protein carrier pump. Bulk transport is a type of active transport in which large amounts of materials are transported through movements of the cell membrane. Here we're talking about endocytosis. This is a process of taking materials into the cell by means of infoldings or pockets of the cell membrane. The sac or pocket then breaks loose from the outer portion of the cell membrane and forms a vacuole or a vesicle in the cytoplasm. Large molecules, clumps of food, and even whole cells can be taken up by endocytosis. And this is actually exocytosis, but endocytosis is actually the reverse of that. So here's what endocytosis looks like. We're taking in maybe a food particle. It makes an enfolding of the cell membrane and closes it in a vesicle. And then the food particles are digested, and then it's removed by endocytosis. Now there are two types of, removed by exocytosis. There are two types of endocytosis. There's phagocytosis, which is the cell eating. It's when large particles are taken into the cell by endocytosis. And then there's penocytosis, the cell drinking, when large particles of liquid materials are taken up by endocytosis. And here we have exocytosis, which is the removal of large amounts of materials from a cell. During exocytosis, the membrane of the vacuole surrounding the material fuses with the cell membrane and forces the contents out of the cell. And this is just an example of exocytosis.